Welcome, everybody. Hi, I want to welcome you all to a very special CDCR seminar this month, and happy Halloween. It is also Diwali, so I'd like to say happy Diwali. And I realized, where's Chitra? I forgot to say happy Diwali this morning. Happy Diwali, Chitra. Happy Diwali, everybody. Or as my 5-year-old nephew said to me this morning, happy Diwali-ween. That was awesome. So welcome and thank you everyone for coming out on a holiday for our very distinguished guest. I'm so honored to have here today Professor Hideho Okada. And I want to tell you a little bit about his distinguished career. Dr. Okada is a professor of neurosurgery at UCSF, University of California at San Francisco, and he's also a member of the Parker Institute of Cancer Immunotherapy. He's trained as a neurosurgeon scientist, and he's been dedicated to understanding the immune mechanisms of brain tumors and developing novel immunotherapeutic strategies for brain tumor patients for over 30 years now. His team was the first to discover cytotoxic T lymphocyte epitopes and glioma-associated antigens. He also found critical roles for the integrin receptor, very late activation antigen, or VLA4, and the chemokine CXCL10 receptor in facilitating the entry of CTLs into the brain tumor site. He has translated these discoveries and developed 8 IND applications that the FDA has approved for early phase clinical trials, including genetically engineered glioma vaccines and T-cell receptor or TCR, or chimeric antigen receptor transduced T-cell therapies in both adult patients and pediatric patients with probably the worst tumor that I can ever imagine that I deal with in my patients is diffuse midline gliomas, or DIPG. And this is an area that really needs some therapeutic breakthroughs, and Dr. Okada certainly has some promising things in the pipeline. One of his vaccine studies evaluated the H3K27M mutation-derived neoantigens. It's actually open at 14 international sites. And he's also developed CARs against epidermal growth factor receptor variant 3, which has been evaluated in patients with glioblastoma. He's also developed a beautiful, technically elegant SYN-notch primed CAR system with Dr. Wendell Lim to overcome antigen heterogeneity as well as off-tumor toxicity and T-cell exhaustion issues. He's really been a pioneer in discovering novel immunoregulatory mechanisms in glioma, especially those that pertain to the tumor immune microenvironment, such as those mediated by myeloid-derived suppressor cells. And we all know here how badly we need to target glioma-associated macrophages. So what I really want to say about Dr. Okada to all of you is that, very often you'll hear the word visionary thrown around, and we use that to describe a lot of people. But to me, Dr. Okada is a true visionary, because visionaries see things in advance of everybody else. And 30 years ago, while he was training as a neurosurgeon in Japan, he recognized very early on that neurosurgery is a very crude tool. And I'll say that today as a neurosurgeon. Neurosurgery remains a very crude tool. And there has to be better ways of addressing the complexity and the heterogeneity of something like a brain tumor. So 30 years ago, Dr. Okada already saw that and recognized that one day there would be molecular neurosurgery. And perhaps that would be putting someone like me out of half of my job, which would be a good thing in the future. So I really do think that Dr. Okada's contributions have served as a guide to many of us in the brain cancer field. And really being able to look at someone who's been able to set the stage for future discoveries, to me, that's the true definition of a visionary. So I'm really thrilled to welcome Dr. Hideho Okada to give this talk today.
Thank you, Sheila. Such a kind introduction. Happy Halloween and happy, excuse me.
Halloween.
Okay, well, so I understand you all work on cancer research, right? But may not be necessarily immunology. And my talk will be focused on brain tumor immunotherapy. And if you're not working on immunotherapy or brain tumors at everyday basis, and if you have any questions, please feel free to stop me and ask questions, because I don't want you to feel like, behind on my talk. I want you to follow my talk as much as you can. And then my laboratory has a lot of things, but all related to tumor immunology, brain tumor immunology. If you know San Francisco geography, this is the city of San Francisco. I can forward you point option. So we have two main campuses. ECSF, the Parnassus campus, which is right here, is more like a traditional campus right next to Golden Gate Park. And about 2010, we have started a new development in the Mission Bay campus, which is on the eastern side. And we are located here, whole brain tumor center located within this building, whole one floor with about 30 PIs are mostly dedicated primarily to brain cancer research. And so I have been so fortunate being in a robust and very collaborative environment. We also have right across our street, we have this stadium for Golden Gate, Warriors, Chase Stadium. And here we have San Francisco Giants Stadium. And right across the street is actually Uber headquarters. But Uber doesn't really matter. Those are stadiums. Although I'm a baseball fan, when we have games, actually when we have events in both centers at the same time, I have a little difficulty going back home. But otherwise, it's a great place to work and live. So if you have an opportunity, please come by. And this is a disclosure slide. Okay, so as I said, I'm going to talk about brain tumor immunology. Why immunology? Why immunotherapy for brain tumors? I was telling people that when I started about 30 years ago, not many people believed in cancer immunotherapy to begin with. People were skeptical. With the immune system, the primary reason is, okay, so cancer has already developed in a patient's body. So that means that patient's immune system has somehow already accepted the tumor. So why are you talking about the rejection of the tumor by immune response? That's kind of weird. And then secondly, even among those people who would believe in cancer immunotherapy, not many of them had believed brain tumor immunotherapy because brain has been traditionally considered immunologically privileged space. Like about 100 years ago, there was an experiment done by inoculating the mouse body, taking like an allogenic, meaning like a graft from another animal, genetically unmatched, and a tissue from another animal, unmatched animal, and then inoculating in the periphery, like under skin. Those things get rejected by the immune response. But if you do the same experiment, inoculating the stuff in the brain, those grafts will not be rejected. So there was an initial observation, hey, brain is immunologically privileged. So when I started talking about the brain tumor immunotherapy, many people said, what are you talking about? And so over time, this has been even updated, but this is a highly nicely summarized sort of cartoon from the review back in 2006. But now we have more knowledge, but the essential component has not changed so much in the sense that, okay, so when we talk about immunological privilege status, what really mediates the immunological privilege? So many people tend to think about, oh, it's blood-brain barrier. Because of the blood-brain barrier, your T cells or B cells won't reach. Well, that's a part of it. I mean, blood-brain barrier certainly makes it a bit more challenging to reach for the immune effector cells to reach the brain parenchyma. But we know by now that it's more about the antigen drainage and the antigen presenting system from the brain parenchyma. I mean, I can talk about it for like 1 hour, but even with the recent rediscovery of dural lymphatics, meaning like from the dura component, there's a good drainage to cervical lymph node, that's for sure. But from the brain parenchyma, which is a brain itself, not the surrounding structure, it is still unclear whether or not effective antigen presenting system exists. What I mean is, okay, the immune system, whenever there's an issue such as a cancer or infection, there's an afferent arm of the immune system, meaning like, okay, antigen presenting cells would show, hey, here's an antigen, here's a bacteria or virus that has antigen, telling to the immune system in the lymph nodes and activating the adaptive immune cells, and those effector cells go back, which I described here as an efferent arm. That functions this way. But in the CNS, central nervous system, what is missing in the summary is the effective antigen presentation from the brain parenchyma. to some extent. And then, so the consequence of death. Okay, so if you are familiar with immunology to some extent, you may know about tolerance mechanism. So immune response is so fascinating in the ways that the immune system will recognize anything that is coming from outside of your body and attack them. But the immune system doesn't attack your own body component because the early phase of T cell development, if T cells are potentially reactive to your own body component or cells, those T cells get eliminated. So therefore, you don't have autoimmunity against your own body component. But autoimmunity condition actually exists against CNS. And so I can talk a lot about brain immunology specifically for 1 hour, but if I pick up one topic as an introduction to try to attract your interest to the topic, I can describe this as a most important interesting topic, iconically interesting. So experimental autoimmune encephalomyelitis, which means it's a long name, but basically, if you grab a mouse, a black 6 mouse, normal mouse, not necessarily fancy transgenic or immunodeficient or gene knockout, anything like that, just a regular mouse, and immunize them with the antigen in the brain, no mutant, just a normal autoantigen from the brain, such as myelin basic protein with management, you induce immune response against the brain. You induce autoimmunity. I do not know any other organs against which you can induce immune response so easily, or there's an autoimmunity. So that tells me a lot of things. So the antigens, because, so again, like I said, the immune response against your own body components is supposed to be eliminated or tolerized, but that's not operating against an antigen in the brain. So interestingly, in human, there's an equivalent condition such as paraneoplastic cerebellar degeneration syndrome. So this is an interesting condition. Patients come to a neurologist's office with the undiagnosed cancer, such as ovarian cancer or breast cancer, that express antigen called CDCR2. So what it is the neuropathology, immunopathology was delineated by Dr. Albert and also Robert Darnell back in 1998 for the first time. So this is a autoimmune condition. So CDR2 antigen is actually expressed in the normal cerebellum, our brain cerebellum. But the immune system doesn't recognize that until the same antigen is overly expressed in the form of cancer outside of the brain, like I said, ovarian cancer, for instance. So that when the patients have the expression of the same antigen overexpressed outside of the brain, the immune system can recognize the antigen as kind of like a non-self immunogenic antigen, and the activated immune response, such as CD8 T cells, can track down to the brain and attack the cells, normal cells express the same antigen. So these observations suggest that if you discover and characterize good antigens, you may be able to elicit good immune responses even against the brain cancer. So there are a lot of caveats: immunosuppression, local and systemic immunosuppression, and myeloid cell mechanism. But just wanted to summarize, bring you some interesting observation about CNS immunology. Brain is not necessarily privileged, but maybe because of the privileged status, that may also provide some advantage of targeting the brain. So today's presentation, I'd like to give you some overview, mainly about 2 topics we are very excited about. One is the CNOTCH. CLAB kindly introduced. This is a novel type of CAR T-cell strategy that should mitigate the current challenges, such as off-tumor toxicities and exhaustion issues. And we just opened the clinical trial of this phase 1 trial. We treated 3 patients safely very recently. We don't have data yet. But another topic I'm going to talk about is the interesting class of new antigen that can be targeted by immunotherapy derived from tumor-specific alternative splicing event. And some of those tumor-specific alternative splicing can induce immune response, T-cell-based immune response that can be potentially utilized for cancer immunotherapy. And I'm going to give also, if I have time, I'm going to give a short snapshot, because talking with some of the faculty members today, I have realized that, many people have different interests, and then some of them are very relevant to the things that we do actually in our lab, but I didn't select for my main topic. So for those things, I try to give like a snapshot, one slide format, and then each of them have either public, the preprint publication or near publication status so that you can find it online more details. So first, I'd like to talk about CNOTCH receptor-based strategy that we developed. So some of the challenges we want to address, we recognize so far, GBM stands for glioblastoma. The heterogeneity, what I mean is, in this case, intratumoral heterogeneity. So there are two layers of heterogeneity that I'd like to talk about. One is inter-patient heterogeneity. If we have 10 patients, and then antigens and genetic landscape are very different across patients. That's inter-patient heterogeneity. On the other hand, here I'd like to address as also an issue of intra-patient tumor heterogeneity, meaning even if you look at the one tumor from the patient within a single mass of tumor, the genetic landscape and immunological landscape can be very different from one side to the other side, or even, we normally take like 10 different core biopsies to understand the heterogeneity of the intratumorally, and they look very different. And then another issue we'd like to address, patients can die. Patients have actually died with CAR T-cell therapy by what we call on-target but off-tumor toxicities. So what I mean by on-target off-tumor means like, okay, so the on-target, so the CAR T cells are actually recognizing the intended target molecule, but the molecule is expressed not only on the tumor cells, but some of the normal tissue, and then even brain sometimes. And so therefore the CAR T cells can target normal critical tissue, thereby affecting the patient's life. And then also an important issue about CAR T therapy for solid cancer particularly is a persistence issue. T cells don't persist very well. Even published papers, persistence is not very long, sometimes like a week, and then oftentimes associated with T cell exhaustion, particularly in the case of CAR T cells. So CAR receptors are the receptor that mediate anti-tumor effect, but it also transmits activation signals to the T cells. And then so those T cells transduced with CAR, they can be continuously activated independent of the antigen recognition. This event can lead to exhaustion of T cells. So this is another issue I want to address. So from a past study that Sheila mentioned kindly, my participation in one of the first GBM CAR T study, working with Carl Jun's group and Marcela Mausno in MGH. So we created anti-EGFRvIII CAR T cells. Anti-EGFRvIII is a tumor-specific antigen. So you may know epidermal growth factor receptor, and the V3 is a variant, mutation, deletion mutation missing exon 2 to 7, but creating a junction, inflamed junction between exon 1 and 8. So it's a deletion mutant uniquely expressed on glioblastoma tissue, although it's only about 10 to 15% of cases. But it can be seen as a very unique cancer-specific antigen. So therefore, we created a CAR and targeted them, and intravenously infused CAR T cells, single dose. And we had an opportunity to evaluate post-infusion tumor tissue in 7 cases. And then 5 of 7 cases, we saw an interesting observation. Actually, T cells actually do come into the tumor site, even though brain is often recognized as immunologically privileged. Like I said, the efferent arm is not as impaired compared to the afferent arm. So T cells do come in and sometimes seen as CD8 cells and a lot of like also CAR negative cells. This is, I think, we try to detect CAR. There's some sensitivity issue, but it is also true that there are a lot of like passenger cells, passenger T cells coming in, even T regulatory type cells, CD25 positive. And it also induces immunoregulatory mechanisms such as IDO1, PDL1. This is reasonable because these molecules are induced by interferon gamma, which can be produced by infused T cells. The point here is though that if you look at EGFRvIII expression status, this graph shows pre-treatment and post-treatment comparison but percentage of EGFR-B3 positive cells. And I want to draw your attention to the fact that it ranges at the baseline. It ranges from 20% to 90-some%. And then you may see some trend, the percentage go down after treatment. However, this heterogeneity level, if you target only EGFR-B3, you may not win the game in terms of potential benefit for patients because it's so heterogeneous. You may miss a lot of cells that do not express the target antigen. And then on the other hand, if you target different classes of antigens, such as in this case, FA2, tyrosine kinase receptor, or IL-13 receptor alpha 2, those are considered tumor-associated antigen, particularly prevalent in gliomas, glioblastomas. Those are non-mutant antigen, but they're quantitatively overexpressed on glioma cells. So if you target them, and if you consider the cells expressing either one of those antigens, your coverage can go up very high, over 95%. 95% may not be enough, but still better. And then, so in terms of those antigens I talked about, FA2 and IL-13 receptor 2, these are attractive tumor-associated, particularly glioma-associated antigens. In the brain, at least, normal brain tissue doesn't seem to express those antigens. Therefore, we've been targeting those antigens in our vaccine approaches ever since, like, 2005 or 2007-ish. And then, however, more recent fine-tuned analysis have identified, developed, and revealed that some of these antigens, FA2 and IL-13 receptor alpha 2, they can be expressed in other normal tissues, especially FA2. Although not expressed in the normal brain, they can be still expressed in many of GI, gastrointestinal tissues, and liver tissue, and so forth. IL-13 receptor alpha 2 seem to be cleaner, but this CAR targeting molecule, the molecule that we use in the CAR called mutant, can cross-react against IL-13 receptor alpha 1 as well, which is ubiquitously expressed on immune cells, for instance. So there are no perfect antigens. So, and I have been so fortunate to work with investigators at UCSF, Paul Roybal and Wendell Lim, because when I joined UCSF, they were about to publish the first paper on synapse receptor as a PhD cell engineering group. They developed this initially to secure like a end circuit type approach, meaning like tumor tissue have to express two antigens to fire for the CAR cells to fire. And so this end circuit requires the cancer cells to express two antigens. So that won't help the issue of heterogeneity. In our case, I'm struggling with the issue with the tumor cells expressing antigen A or antigen B. There aren't many. There aren't good antigens that are, ideal tumor antigens should be the ones that are uniformly expressed on tumor cells, but not expressed in normal tissue. But there's no such an antigen. But when I saw this concept, I thought this would be a great tool to mitigate on target of tumor toxicities and even tumor heterogeneity. So the synapse receptor is named synthetic notch because the intracellular transmembrane domain comes from the notch receptor, but all the other portions are synthetic. Say you can make a receptor that will recognize surface antigen, kind of like a CAR, but this is constitutively expressed and transmit a signal. And the notch receptor, by nature, upon activation or upon recognition of the ligand, the gamma secretase or ADAM10 pathway comes in and cleaves the intracellular domain. This intracellular domain translocates itself to the nucleus and express other genes, secondary genes. So this portion can be synthetic. So they made a way in the way that they can have like your gene of interest expressed by the recognition of antigen here. So you can create this circuit. And then again, I thought this would be a great tool to mitigate the issues that we face. The dilemma is that we have no perfect antigens. The antigens can be classified either antigen type A, that would be tumor specific, or brain-specific, but not uniformly expressed on tumor. But antigen B can be glioma-associated antigen and may be more broadly expressed, but not necessarily tumor-specific. And then so the left side of the cartoon shows like a traditional CAR T therapy, like I showed as an example, EGFR-V3 targeting CAR as an example. So if you target antigen A in this case, you can eliminate the tumor cells that express antigen A, but all the other antigen A negative cells remain intact and they come back as a recurrent tumor. On the other hand, when we developed CNOT originally, okay, so let's use, the idea was to use antigen A as a brain-specific or antigen tumor-specific signal that should give us signal to induce another CAR that would recognize tumor antigen more broadly. So in this case, antigen A serves as the priming signal for this circuit. Then T cells would express another CAR against antigen B in this case. And antigen B is the one that is more uniformly expressed so that we can maximize the anti-tumor effect. On the other hand, the downside of antigen B is that they can be expressed in normal tissue like this. But because of the absence of priming signal antigen A in this case, there will be no priming, there will be no toxicity. That was a theory. So when we first tested our hypothesis using EGFR-V3 as a priming antigen in this case, that will induce the antigen, a chimeric antigen receptor against FA2 and IL-13 receptor alpha-2. This is an OR circuit, meaning like tumor cells needs to express either one of the antigens to be killed. And then when we tested this in vitro, if you have 10% of EGFR-V3 positive cells in the culture, although all the other cells are negative for EGFR-V3, they express this CAR receptor, CAR target antigens, and you can eliminate an entire cell population. And then if you move to, when we move to in vivo study using patient-derived naturally heterogeneous PDX that express about 50% of cells express EGFR-V3 and tested it intravenous injection, single injection of this circuit cells. So reproducibly, we see a long-term sustaining effect, complete remission in most cases. And then when we tested a conventional CAR T cells, recognizing the same antigen, IL-13 receptor alpha-2 FA2, and even though those T cells can kill the target tumor GBM6 cells in vitro very well, when we bring to orthotopic model in the brain and animals, they don't kill, they don't do anything. And then EGFR-V3 CAR works some good extent, but the tumor comes back as a EGFR-V3 negative tissue. And the part of the mechanisms that we've identified was a difference in the T cell phenotype. So here, histogram shows T cells, juvenile nature of T cells by looking at CD45 RA and CD62L, which is a marker for central memory type cells. So if you look at the double positive cells, those are the type of T cells that are more like T stem cells or central memory type cells. They provide longer, they have longer longevity and persistence in vivo. However, if you transduce the same donor-derived T cells with constitutive traditional CAR, they differentiate into more like effector phenotypes. They have short living life and they are more exhausted. And then CNOT CAR circuit, reproducibly with different circuit, we see very similar phenotype as untransduced juvenile T cells. So this is, we think one of the key mechanisms how CNOT CAR T cells better persist. So I just told you about EGFR-V3 primed signal system, but the EGFR-V3 is present only in about 10 to 15% of adult GBM patients. What about other patients? Or in fact, none of the pediatric brain tumor patients express EGFR-V3. We care a lot about pediatric population because pediatric brain tumor is the most common cancer-related mortality morbidity. Now, so what to do? And then we came up with a second idea, which is to prime this circuit with brain-specific antigen. And so instead of EGFR-V3 as a cancer-specific cue, what if you prime the circuit with brain-specific antigen? It should be safe because there are no CAR antigens expressed in the normal brain. And even though normal tissues such as GI tissue express CAR target antigen, but because of the absence of a priming signal, which is a neuron or brain-specific signal, the CAR shouldn't be primed in non-CNS organs. Even if those T cells that are primed in the brain migrate out, that we know that the expression of a CAR after priming is transient. The half-life is about 12 hours or 10 hours or 12 hours. So even if they migrate out from the brain, the expression should attenuate. And then the toxicity should be minimum. So this is our idea. And then we tested several different brain-specific antigens and found that the BCAM, BRAVCAM, which is a proteoglycan, highly expressed, but restrictively expressed by brain tissues. This function as a best priming antigen for us. So here, I already told you about E-sync circuit. And the right side, this is B-sync. So E-sync standing for EGFRB3 primed, and second generation BCAM primed, we call it B-sync. Okay. So the B-sync circuit is amazing. Also, we have a immunocompetent mouse version of the same system that works very, very robustly. And in this case, we tested a similarly orthotopic brain tumor PDX model. The single dose infusion leads to a long-term remission. And we have clinical trial candidate vector here. But again, reproducibly constitutive traditional type of CAR recognizing same antigen didn't work. And this single infusion of B-sync T cells mediate long-term protection effect, as indicated here. We re-challenge the same animals with the new tumor cells after 70-some days after the initial infusion, and they are protected. And then we have a lot of data because we submitted IND already, an open IND, but part of the hypothesis that these cells are primed only in the brain and mediate anti-tumor effect only in the brain. So to prove that, we conducted one experiment in that animals have the tumor in the brain. This is BCAM positive. And then we inoculated a BCAM knockout version of the same cell line as a subcutaneous tumor. So the subcutaneous tumor grows, and regardless of the treatment. So the B-sync CAR cells IV infusion didn't affect any part of the tumor growth, while the same treatment completely controlled the growth of the tumor in the brain. And then so we are also fascinated by their amazing ability to track traffic to the brain. So these are flow cytometry taken several days after the infusion. And I want to draw your attention to this gated population, which are CD45 and CD3, meaning like human T cells infused into these animals. Obviously, you see those T cells, a lot of cells in the spleen. So this is the case in the B-sync T cells and controlled similarly activated T cells. We also tested constitutive CAR T cells. But if you look at the brain, the B-sync cells have an amazing ability to home to the brain, but similarly activated control cells didn't come to the brain. And so in this case, the GFP is used as a surrogate marker for the priming of the circuit. And as you expected, the cells are primed only in the brain, but not so many in other organs. And some of those brain cells express CD103, which has been implicated as a brain tissue resident memory cells. Those are not primed, but interestingly, they persist for a long time. They sort of come back and forth in terms of the priming state versus resting state. So this is my translational team, a multidisciplinary team that we are working together to develop clinical trials. And for clinical trial consideration, there are lots to consider. I'm not going to touch upon too much details because this is, I understand the audience is pretty much more like geared towards basic science aspect, but there are a lot of things to clinical consideration that needs to be put. And so we recently published a review paper in Neuro-Oncology to talk about all those important considerations and clinical trial design. And also in the field of CAR T cells for brain tumors, a lot of investigators have been using intraventricular delivery approach. So there are ventricles in the brain filled with cerebral spinal fluid, CSF. There's a device called Omoya reservoir whereby you can directly infuse your T cells here, or IV infusion approach, systemic. And then with that, the T cells would circulate the systemic blood circulation. The many T cells can have to go through the lung because this is the first organ that IV transfer cells go through. And so there are some indications, interesting research that lung tissue can actually promote the phenotype of T cell that in the way T cells can more effectively traffic to the brain. There's an interesting autoimmunity research like that. We still do not know what happens in the setting of CAR T cells. But please look at our review to talk about mechanism and pros and cons that we discussed about different delivery approaches. So we just recently opened our study using EGFR-B3 as a priming antigen, supported by National Cancer Institute and also SAM, California Institute for Regeneration Medicine. We have a sequential two-cohort design. The cohort number 1, as we discussed with some of you earlier today, we enrolled patients who are recently diagnosed with glioblastoma, who would undergo maximally safe resection, surgical resection, and radiation and temozolomide concurrent chemotherapy. And we take lymphocytes by leukapheresis and infuse them as a single infusion. And our primary endpoint is safety evaluation. And then we have 2 dose escalation design. And with the highest safe dose, we will move to cohort 2, in which we will ask more scientific and biologically interesting question as to whether those infused T cells can come to the tumor and primed in the way we expected. So for that, we ask patients who are clinically indicated for re-resection surgery for their recurrent tumor. And before that, we infuse T cells and look at the tumor tissue. So we concurrently developing pediatric version of the trial with the Sabine Mueller and her pediatric group. In that, we are actually planning to combine, this schema may not be, here you go. We plan to combine IV infusion, but also ICV, intracerebral ventricular infusion. So this trial is also concurrently being developed. So this portion, I would like to summarize that single IV infusion of E-Sync or B-Sync cells leads to sustained complete remission of the aggressive intracerebral GBM PDX model. We also have data similarly effective using 2 different DMG, diffuse midline glioma PDX model that we didn't have time to talk about it. But observations associated with restricted priming of the CAR in CNS brain and the remarkable persistence of IV-infused cells in the CNS tissue. We also have, as I shown as a histology, flow cytometry histogram, we see stem memory-like phenotype of those T cells that should be, they can be contributing to the good persistence of the effect. So the trials, we treated 3 patients as of yesterday, 2 days ago, safely received those infusions who are working on correlative studies as we speak. And the B-Sync version phase 1 trials are being developed for adult and pediatric glioma patients. So I'm going to switch my gear a little bit, second topic, which is to talk about a little bit more basic science aspect, discovery phase story, talking about cancer-specific alternative splicing as a source of neoantigens that can be targeted by immunotherapy. For this, I have been so fortunate to collaborate with Dr. Joe Costello. As many of you may know, he's an international expert, one of the most respected when it comes to brain tumor genomics. And so we wanted to talk about immunogenomics. It's a natural collaboration, multidisciplinary collaboration. And our co-mentor, graduate student, Darwin Kwok, who recently defended a PhD on this project. Now he's a medical student in UCSF. So he did kind of opposite way. He did a 5-6 year PhD program, not associated with MD program. So unlike typical MD PhD program, he did a PhD first and completed it now. And then he applied for medical school, and he is now in MD school at UCSF. Nick Stevens and Chris Klebanov is a Memorial Sloan Kettering cancer immunology specialist, particularly when it comes to T-cell receptor, and Inaki is postdoc for this. So this has been a fantastic collaboration to try to overcome the antigenicity issue. The idea is to try to find a different source of cancer-specific antigens than somatic mutations. So this is often cited in terms of numbers of somatic mutations found in different types of cancers. And if you look at glioblastomas or pediatric brain tumors, they belong to medulloblastoma right here. So they kind of belong to the type of cancers that have low or even low medium numbers of mutations. So if you expect immunogenic mutations, so very careful analysis have shown that only 1 or 2% of non-synonymous mutations can be immunogenic. So even if you find 100 mutations in the cancer tissue, the chance of having one really immunogenic antigen may not be very high. So we definitely need to think about alternative source of antigens. And also we want to address intratumoral heterogeneity, as I discussed earlier. So how the question relates to how often we see alternative splicing events in a cancer-specific manner, whether it is heterogeneous within the tumor, and whether we can target them by immunotherapy approaches. So our initial hypothesis was our original interest was centered around IDH mutant tumors. IDH stands for isocitrate dehydrogenase. Excuse me, I should have had a title slide somewhere, title button here, but I missed it. And then so isocitrate dehydrogenase is a TCR enzyme. There's a unique mutation in a subset of gliomas that caused the global methylation, hypermethylation in histone and the DNA level. This causes a massive, significant epigenetic changes. And we hypothesized because of that, these tumors would likely to have a unique and very high frequency of alternative splicing, because splicing events can be also modified by the epigenetic changes. So to address this question, we started looking at the TCGA database in silico, and then we selected non-annotated alternative splicing isoforms based on TCGA. And then we wanted to select cancer-specific ones. So we looked at the GTEx and eliminated the ones that are present more than 1% based on GTEx. And we also wanted to have public antigens rather than private antigens, meaning like as an antigen, we want to see the same alternative splicing in multiple patients in the same type of cancer rather than patient-specific. And we also wanted to understand intratumoral heterogeneity. As I may have mentioned, in each tumor case, we tend to reject multiple cores, up to 10 different cores, and analyze separately, genomically and immunologically, so that we can understand immunological landscape in a heterogeneous tumor. And then we run our platform and a pipeline to predict immune epitopes, and we validate the immune response in the wet laboratory system. So kind of summary, we have a lot of data in this preprint, and also we just resubmitted our manuscript. And then Darwin actually found that actually, as we hypothesized, IDH mutant tumors, like the yellow ones and the red ones, they have actually much significantly higher numbers of neo-junctions based on the pipeline analysis. And not only that, if we look at the utilization expression level of splicing factors, there are clearly distinct patterns of splicing factor expression level. There are a group of splicing factors that are upregulated in those tumors, but also ones downregulated in these tumors. So the mechanism is not just simply uniform or simple. There must be a lot of multiple mechanisms involved in the overall landscape creation and the creation of alternative splicing events. But we want to particularly look at the IDH mutant tumors, why IDH mutant tumors have higher number of neojunctions. And then in this case, we found that one of the splicing factors here, excuse me, I can't, how can I get rid of this? Hide?
Yeah, I think that's good.
Okay, let's try this. Come on. Hello. All right. Yeah. And then so this splicing factor is actually mapped on chromosome 1p, which is deleted in these tumors. So therefore, the low expression of this splicing factor, because of the genetic deletion, is associated with, in this case, higher number of alternative splicing of particular target neojunctions. And experimentally, using GBM cells that do not have this gene deletion, if you knock out this splicing factor by either shRNA or CRISPR-I experiment, you increase the number of expression of neojunctions. So we have some mechanistic insight how those neojunctions can be generated. And also we looked at the heterogeneity of the tumor of expression within the tumor. So here I have two layers of information to tell you. Okay, so the y-axis shows percentage of the presence of a neojunction, and then x-axis, these are the total, each column indicates each neojunction. So we have looked at probably hundreds of different neojunctions, and the ones on the left side, they have higher frequency. So for these ones have nearly 100% of frequency across patients. But the color code, this is the second layer of information I want to draw your attention to. So the red ones are likely to be tumor-wide, meaning like when we look at the 10 different tumor cores, you see the expression everywhere. So color-coded, the red ones are most presenting across the tumor. The yellow ones are somewhat heterogeneous, and the green ones are highly heterogeneous, meaning like we only see the expression of this in one or two cores. So there's a different degrees of frequency and heterogeneity, but we want to also evaluate potential immunogenicity of them. So for the antigen to be presented by MHC so that T cells can recognize the antigens and epitope, that alternative splicing product has to be presented by the MHC. So there are different programs out there available for us. Some of them are more centered, prediction is centered around MHC, major histocompatibility binding. And the other programs are more heavily sort of programmed based on the pre-processing pathway of antigen. So we wanted to combine those multiple different programs to predict most likely promising candidates. So we applied those two different algorithms and selected ones that are scored very highly by two different algorithms, and selected them and tested them. So in this case, we tested one candidate derived from antigen called GNS alternative splicing. And we had initial screening system using COS7 cells transduced with HLA and the target neojunction mRNA. And the readout can be, in this case, T cells, the T cell line, actually, JACA T cells transduced with the TCR. Also, we cloned those TCR against each of those antigens. And the important figure is here. So the flow cytometry shows NFAT activation, meaning, okay, these cells are recognizing the antigen presented by MHC. And if you go with the short antigen, of course, they can be easily presented because there's no endogenous processing system involved in it. So this is more like a positive control. This column is more like a positive control. But the important portion is here when cells are transfected with full-length RNA, including the neojunction portion. This requires the internal processing in the way we expected so that the epitope can be presented on the surface of MHC. That was the case in both of the candidates in this case. So this portion is highly sort of immunogenic, and this portion is more like a physical validation of an antigen. So there's another way to validate the presence of antigen epitope on the surface of MHC, which is do it more physically instead of functional. I mean, the previous slide, I showed the data using T cells. We asked T cells to tell us whether antigen is presented. Here, more like a physical approach, directly stripping the antigen from the surface of the cells and detect antigen in the mass spec analysis. And in fact, my collaborator used memorial Sloan Kettering cancer. We actually blinded them. We didn't tell them about the epitope. We intentionally kept them blind and asked them to tell us what they see. And interestingly, they came up with the same epitope that we identified, a T-cell approach. So it was very nice validation, independent validation. They even found the same antigen epitope present in the glioblastoma cell line, so even a naturally expressed epitope can be presented. And then eventually we tested the whether or not TCR, T-cell receptor transduced T cells can kill those tumor cells that express the neojunction target. And the busy graphic, excuse me for that. So there's a good level of killing, and in some of the cells expressing the target, and a specificity to show the specificity of the response against with the HLA-A2, because HLA-A2 in this case is the one MHC molecule that expressing presenting the antigen. If you don't have HLA, this is a negative cell line for HLA-A2, you don't get the killing. But if you transfect the same cells with HLA, now you get killing. So, so this study is currently in evaluation in second round review, and hopefully we will see publication. And important implication, if we go through, this may allow us to target different class of antigens in the future immunotherapies. And also we have a lot of data. If you go to the preprint version, we've looked at 11 different cancer types. So this is not about glioma. I need to squeeze down into 10, 15 minutes format, but if you look at the preprint, you may find similar, very interesting outcomes for the type of cancer that you are most interested in. Briefly, just a snapshot. So Jonathan and I talked about earlier today, thrombospondin as an interesting molecule. Here, just a one slide snapshot. So we looked at, we got interested in the potential interaction between neuronal connection and cancer cells and the immune system. So there's been a lot of study recently indicating that neuronal transmission pathway signaling can promote cancer cell growth in the brain, and the brain cancer signaling can also impact the neuronal transmission levels. So it was a dialogue between tumor cells and neuronal cells. But when we looked at the data of those data, differentiating high functional frequency, high functional connectivity area, that is a tumor area where the neuronal activity is very high versus tumor area in the same patient where the neuronal connectivity is relatively low. We saw striking this difference. In an unbiased manner, 6 most downregulated pathways in high functionally connected region was actually immune related. And this was the case in myeloid cells, tumor cells, and even lymphoid cells. So we have a bunch of animal model validation of the mechanism. So this paper is now under revision. We just resubmitted the revised version, but this is available as a preprint. So if somebody is interested in thrombospondin, because this difference seems to be mediated by thrombospondin. If you knock out thrombospondin in the syngeneic glioma model, you recapitulate the same thing. So I don't have enough time to talk about it, but please look at this preprint paper for your interest. We are also interested in myeloid cells, tumor infiltrating myeloid cells, and those are mostly immunosuppressive macrophages, but can we turn them into more like functional dendritic cells? What if you give them like a master regulator of dendritic cell differentiation, such as IRF8? So we did that by using retroviral, conditionally replicating retroviral vector. Interestingly, although retroviral vector infects only in the dividing cells, in the tumor microenvironment, many myeloid cells are actually proliferating within the tumor. So thereby they are susceptible to the transfection with retroviruses. And when we do that, we saw that improvement of antigen presenting function, as I shown here, T cells can proliferate much better in response to IRF8 transfected disease in the tumor. And so this is kind of key message. So we recently published this in Neurological. If you're interested in myeloid cells, please look at this paper. And if you look at if you're interested in metabolism, we have an interesting study in that hypoxic condition, brain tumor, particularly severely hypoxic. So we want to find a condition where that allows T cells to function even in hypoxic condition. So in this paper, Ryusuke Hatae, a neurosurgeon from Japan, we identified pre-treatment of CAR T cells with metformin and rapamycin really promote the T cell phenotype, and T cells are more resistant to hypoxic condition and mediate a better anti-tumor response. And when it comes to clinical trials, some of you I discussed about our interest in vaccine. We are most interested in like a preemptive vaccine against cancer recurrence and progression. As I mentioned, IDH mutant tumors, gliomas, tend to affect young adults. They are slow-growing tumor initially for a number of years, but they eventually recur, and most patients succumb to the disease. So can we preemptively vaccinate those patients? We have had a two-arm neoadjuvant study in which we evaluate the impact of GBM6-related vaccine. Here, the idea was to use a GBM-derived antigen treating lower grade glioma patients. So the idea was to mount immune responses against high grade glioma related antigens in the patients who have lower grade glioma so that vaccine can be preemptive. So we have learned a lot from this study. Some of the T cells responsible to this vaccine can be found in the brain tumor site. Okay, so sorry, I may have go beyond my expected time, allotted time. But this is the most important slide. This amazing team at the UCSF, my laboratory members, current members, and past members, and collaborators within the UCSF group, multiple groups, and also funding industries and funding agencies. But most importantly, we really thank our patients who come on a visit, participate in our clinical trials and studies. Okay, so thank you so much.
Thank you, Hideho. That was a tour de force. That's so much technical elegance. You must be exhausted just to review all of this. So questions, open for questions. I'm sure all of our brains are full of questions. Lara.
Thank you so much for the presentation. I'm curious on the first part of your talk.
Sure.
You were mentioning about the clinical trial for diffuse midline glioma. What is the reasoning of using both intravenous and a follow-up with intraventricular?
Yeah. Great question. That's because we see pros and cons of both approaches. In general, intravenously infused CAR T cells tend to persist longer. And that makes sense because T cells are in the place they're supposed to be in, blood circulation. They can reach lymph nodes, they can reside in lymph nodes. And on the other hand, CSF is cerebral spinal fluid, is essentially saline. And it's like, saline. The protein concentration, for instance, in CSF is less than 1% of what you see in serum. And then there are no nutrients or components that should promote T cell survival or proliferation. So as a matter of fact, usually the many studies that focused on ICV delivery of T cells, they don't show very good persistence. On the other hand, the CSF, although CSF compartment is still separate from brain parenchyma, it's separated by glia limitants. There's a mechanical structure barrier between, but there are some studies have shown really direct migration of T cells from a CSF space to tumor. So there can be more direct approach in the tumor that is localized in the CNS. So we see pros and cons. And so we want to maximize the chance for patients.
Jonathan.
Hideho, I know that you're not talking about thrombospondin today, but I can't help but notice that the gene expression profile of your myeloid cells looks a lot like the profile we see with gamma delta T cells co-cultured with GBM. I mean,
Really?
that down to the
Yeah.
exact pathways that are upregulated. I'm just wondering, do you know if you just take myeloid cells and expose them to thrombospondin alone,
Mhm.
will you get the same pathway, or
No.
is that's just more what's happening inside the tumor?
So that is the case. So we did extensive studies to assess the direct impact of strong responding one on immune cells, including myeloid cells and T cells. We didn't see this.
Right.
So therefore, we came up with a secondary hypothesis that thrombosponding from the tumor, maybe we know for sure that enhances and activates the neuronal connectivity. And enhanced neuronal connectivity seems to show induced immunological changes. That's been shown by pharmacological inhibition of glutamatergic pathway in this paper and in vitro experiments.
Okay.
So
And do you know if these myeloid cells, and again, I haven't read your paper, so I'm going to read it tonight, but are they tumor supporting, these myeloid cells?
yeah,
Is that phenotype a tumor supportive phenotype?
yes, yes. And then tumor supportive and also T cell suppressing. But that phenotype can be reversed by knocking out the thrombospondin expression
tumor
in the
cell.
tumor cells.
Katie,
Beautiful work. I just was curious about the neojunction
Yes.
approach and the examples that you showed. From an evolutionary perspective, how critical are these neojunctions for the tumors, and how
Yeah.
easy do you think it might be for these cells just to turn off this particular splicing?
Right, that's something, that's a great question. We are still working on it. Well, one of the neojunctions we recently discussed with a investigator focusing on DIPG oncogenesis. And this is still sort of, I can't talk about too much detail, but this person has independently found one of these alternative splicing as a potentially critical molecule for oncogenesis of midline glioma. So we are interested in that aspect.
Ali, and then Eugen.
In the experiment that you injected tumor both in the brain and in sub-Q, and then you injected IV, it was interesting that you saw more cells going to the brain compared to that. Can you comment why?
Yeah, well, thank you. So so in the last 20 years, my laboratory has been studying about factors that would promote T cell forming to the brain, such as integrins receptors and chemokine signals. We see those in our cells, but also we see the same amount, same level of expression in constitutively transduced CAR T cells, conventional T cells. So we still do not see a differentiating factor because again, we see those factors that we knew of in both traditional CAR T and Synapse CAR T. Something I can tell is that I think it's a combination of homing and persistence in the brain and potentially proliferation. That's why we see as an outcome higher numbers, because higher numbers in the brain can be attributable to those events, each of those events, right? So
Thank you. Yeah, if it goes to the brain and proliferate, that's fine. But what
Yeah.
drive them at the first place to home to the brain, but not to the sub-Q?
No, they do home to sub-Q as well. I want to show you. I think you were talking about this data set. Thank you. Well, I would say conventional CAR T cells fail to home to the brain. On the other hand, the Synapse CAR T cells, here, this is the data set you're asking about. So presence of T cells, you can see a lot in the brain. There are some T cells in the flank tumor here, but the striking differences between the group looking at the brain tissue. So I would say that even B sync cells, they migrate to the spleen. We do find a lot of cells in the lung too, and we are actually analyzing even after the same infusion, the population of cells that are found in the brain versus found in the lung we did single cell analysis and we're trying to find the factors that are mediating the difference. We're working on it.
Fusion.
Yeah, I just noticed that, because from your original SynNotch paper, you used a different CNS marker, MOG, and you switched. Is there a reason why you switched to?
Oh, in the 2021 paper, we described MOG,
Yes.
mining oligodendrocyte glycoprotein. Thank you for your attention. It's interesting, yes, that was included in the original paper. We tested that. Interestingly, MOG is not expressed on tumor cells at all. That molecule is almost restrictedly expressed on oligodendrocytes. So it was so interesting to see that tumor regulation can be mediated by that signaling coming from oligodendrocyte despite absence of expression on tumor cells themselves. And so it was a conceptually very novel in that aspect, but therapeutically, it's a BCAN is much more robust and applicable for different tumor locations, even including brainstem. Yeah, so that's why we got more interested in BCAN.
Thank you for the amazing talk, actually. For the splicing, because we observe functionally important and related splicing event actually regulates by one key splicing factor or sometimes coupled by a few positive and negative splicing factor. I wonder whether, like in the case you showing as an RPD2, do you also identify other potential splicing regulator regulating the splicing event, or you actually, in addition to one splicing event, you may see a correlated networks of multiple splicing events?
Yeah, so yes, question is about specific roles of splicing factors creating new junctions. So I have to say that, so we profiled the splicing factor expression levels and we see differences, but we didn't characterize every one of them precisely. It's like the demonstration is with the one or two molecules that we have in the manuscript that can be just a really tip of the iceberg. So we can find more by investigating, but it's such a global and probably complicated multiple layers of events for each organs new junction creation event, because sometimes we see no correlation by knocking out, no impact sometimes. So therefore, single molecule contribution may be limited. Must be multiple components. We see something like that too. And so, yeah, we still need to learn more comprehensive pictures.
Any other questions for Dr. Okada? If not, then please help me thank him for a wonderful talk.